blood group system, we have other clinically significant or important blood group antigens that we should also pick interest in. And these are the Kell, the Duffy, the Kid, Diego, MNS, Lewis. I just mentioned this because at a certain point in our protocol, we actually advise to do antibody screening. And if we are doing antibody screening, that means the antibodies that we'll be looking for are actually these important ones. So I just put this one slide to show you which antibodies would be interested in case your facility starts doing antibody screening. So let's talk about the D antigen and an antibody. So we all know that the rhesus D, capital D, can generate a very strong immune reaction, and that produces anti-D in a rhesus negative person. Produces anti-D in a rhesus negative person. That means, should you, by any mistake or because of your method or protocol, introduce a rhesus D positive blood into a patient who is rhesus negative, you are going to make that patient produce an antibodies against the capital D, which will eventually cause problems and transfusion reactions at a later stage. So the anti-D which is formed as a result of stimulation from the exposure through transfusion or pregnancy or solid organ transplantation can actually cause transfusion reaction. So there are basically three ways you can introduce this or produce a strong immune reaction. So it is either through blood transfusion, pregnancy, or when you're doing organ transplantation. So unlike the ABO blood group system, the rhesus D negative people do not actually have naturally occurring anti-D antibody. So the first exposure is very, very important because it will introduce the antigen into them. And then the first time you give, it will not really be risky because you are just initiating the sensitization of the red cells in these patients. So everything will look normal. Even when you do your cross-match on the tile, everything will look normal, but not knowing that you're actually introducing or initiating the sensitization of the red cells in this patient unknowingly. So the probability that the anti-D antibody is produced after the first exposure is very, very high. And we are saying the D antigen is highly immunogenic, meaning it can actually produce a strong immunity against the D antigen. So if anti-D is formed, this poses a high risk of a transfusion reaction if D red cells are received or are given to the patient. So there can either be hemolytic disease of the newborn or fetus in a later pregnancy, which will eventually cause either miscarriages or even a child being born and then it dies immediately or after some time. So this is the reason why patients and donors must be matched for rhesus D, and it should actually also include the antibody screening where possible. But in our setting where you cannot do antibody screening, just match the rhesus D for the patient during, and the prenatal testing. Because we see mothers who come to your facilities and some of them turn out to be rhesus negative, but we don't proceed to do other investigations. And I want you to maybe from after this presentation, we start initiating some steps just beyond stopping on rhesus negative, and then we report. So what do we use to actually determine the rhesus factor? We have already commercially prepared anti-sera, we call anti-D, readily available out there. But the challenge with this is that most of our facilities also don't validate the quality of this or the potency of this anti-D. Sometimes, I remember at some point in our practice here, we received a batch of anti-D which was not passing our criteria of validation criteria. So when we sent them back to NMS, they redistributed to other facilities. It could actually include yours. And many of our colleagues started reporting rhesus negative, yet actually was a false rhesus negative as a result of the poor quality anti-D which was supplied to them. So we use anti-D to determine our rhesus factor. So which test methods do we use? I would encourage us to use the tube method and the microplate. The microplate is basically at the blood bank settings. In hospitals, you can comfortably use the tubes. I have intentionally decided to omit the tile there for one very good reason, that in the event that you actually supplied with the poor quality anti-D and then you use the tile, you might miss out very many things. So try to insist on the tube method if you can, but if you can't, use the tile. But for the weak D that we'll later on discuss more, I would not encourage anyone to use the tile. So it's basically the tube method. So how is it done anyway? So you have your anti-D reagent, which is commercially prepared, and then you have your purple top sample. So the thing is, you have to first centrifuge the sample and then remove your plasma, unless you're doing indirect, reverse. But if you're just doing your direct, so you have to remove your plasma, wash the cells, and then make your 5% red cells, the 25% red cell suspension if you're using the tile, and then you mix with your anti-D and you get your results. But if it is a tube, you're emphasizing 5%, not 25. So it is the anti-D mixed with the washed red cells. So it should be able to give you a positive reaction or an agglutination, and then a negative reaction if it is negative or if the antigens are not there. Unfortunately, the D antigens are a bit complicated. They are a bit complicated. Why are they a bit complicated? Because the first point here is the D antigen is highly immunogenic. They stimulate immune response. However, all human red blood cells have the RHCE protein, which carry this capital C, small c, capital E, small e antigens. In this case, I'm talking about whether you're SSD positive or SSD negative, you have the RHCE protein. That cuts across, everyone has. It might be small e, capital E, or capital C, small c, but all of us have, irrespective of your RSA status. Negative or positive, we all have that protein. Now, only the RSD positive individuals, red blood cells, carry the RSD protein. And they differ from the RHCE, which all of us have, in 35 amino acids. So this is what makes a difference. This is what makes a difference between the CE and then the RHD. So this major difference causes strong stimulation of the immune system of the D negative person if is given blood which is RSD positive. So this is the idea. We have the RH proteins, okay, with the RHD, and then we have the RHCE. And I'm saying every individual who is RSD negative or positive will have the antigen for C and E. Okay, now the problem comes in some individuals who are RSD negative. And how do they even become RSD negative in the first place? The RSD negative comes as a result of the deletion of the gene of RSD during the process of protein synthesis. So this comes, it can actually be, I'm giving you the reasons for the deletion at a later time. That what can cause the deletion of RSD anyway? One is mutations in the RSD gene. It might be small insertions or even a larger mutation. And sometimes there can be altered amino acid sequences in the D protein. And once it is altered, we lose the D, and it will never be expressed anywhere. Then the third point here is unequal crossing over during meiosis. The RHD, that is the RSD, capital D, and then the RHCE genes are located very close to each other on chromosome 1p36. And because they're very close to each other, they usually share very high sequence homology. So any misalignment causes unequal recombination, hence the deletion of the what? Of the RHD gene, and eventually remain with only the CE. D will not be there. And also sometimes there can be what we call gene conversations sequences of the RHCE and then the RHD. Should there be any mix-up there, we lose the D antigen. I mean the D gene, which will eventually supposed to be expressed as the antigen at a later stage. So this is how we end up with some people being rhesus negative and others rhesus positive. Then the question here is, we meet some of these challenging questions, that if both parents are rhesus D positive, mother and father rhesus D positive, is there any chance that they can have a child who is rhesus negative? And I'm saying yes. There is a higher chance that there is actually a higher chance that you can have rhesus negative children when both parents are rhesus D positive, for the reasons that I've listed here. And remember one thing, that the rhesus D gene is not sex-linked. It is not. This is an autosomal chromosome, okay, which is chromosome 1. So anything can happen. Maybe because of the mutation, you end up having some children with rhesus negative and others are rhesus D positive. So we shouldn't really start condemning some mothers out there that they went and did what because you're trying to relate the rhesus D factor to inheritance. So its inheritance is very, very independent of the sex-linked genes. This is an autosomal what? Chromosome. Just like how the inheritance of the mitochondrial DNA is, that some children actually inherit the mitochondrial DNA from their mothers, not necessarily the father. And if the mitochondrial DNA carries some defect, so most of the children will actually have the defect because they inherited from their mothers, but they are not necessarily sex-linked. So there are about 4 scenarios in this case. If you see red blood cell which is naked like that without any D antigen, on the red cell surface, that is a true rhesus negative individual or rhesus negative patient. No antigen on the red cell membrane, it is plain. But in the event that you see the red cell membrane covered with the D antigens, this now becomes a true rhesus D positive individual or patient. Because what is responsible for determining this is either the presence of the D antigen or absence of D antigen. Absence of D antigen results in rhesus negative. Presence of D antigen on the red cell membrane results into rhesus D positive. Now here comes another trick, that sometimes you may have the rhesus D antigen on the red cell membrane, but they are very few in number. And this brings about the quantitative difference, because the few rhesus D antigen on the red cell membrane are not enough. They don't provide enough binding sites when you mix them with the commercially prepared anti-D. So in most cases, we end up missing this reaction. And these individuals are called the weak D individuals. Okay? Then the other scenario is that, yes, this person has very many D antigen on the red cell membrane. But the problem with them is that the very many D antigens do not provide normal binding sites for the reactions with the commercially prepared anti-D. So when you mix them with your anti-D, you are still going to miss out the reaction, because very few of those many D antigens have normal binding sites. The rest are abnormal. So we call this qualitative difference. So the quality of the binding sites on these individuals is very poor. So in most cases, you end up reporting them as rhesus negative. So what do we do in our facilities in most cases? We do not differentiate between the true rhesus negative and then the weak D and the partial D. But in most cases, what we can do is report both the weak D and the partial D as rhesus negative. And we are going to see the dangers of this at a later stage. The dangers of reporting weak D and partial D as rhesus negative, because we don't go ahead and confirm this. Yeah, so let's look at the D variants, which I've already mentioned, that the weak D, they have fewer than normal antibody binding sites per cell. So it is a quantitative variation. And then the partial D have abnormal D antigen. One or more epitopes are missing. which brings about the qualitative variation. So the quality of the binding site is very poor for the partial D, while for the weak D, it is the number of the normal binding sites. So let's look at the overview of weak D generally. Back in the days in the old literature, we were calling this DU. So it was formerly called DU, but in the new literature, we now call it weak D. So the weak D cells are classified as weak D type 1, 2, 3, and can even be more, okay? Depending on the number of the antigens per cell. And this type 1, 2, 3 cannot be established in your facilities, ladies and gentlemen. This is done at a molecular level, okay? Yeah, so some weak Ds are not agglutinated using your standard what? A direct agglutination test. So they seem to appear as rhesus negative. And some weak Ds are actually immunogenic. I mentioned this earlier. So they will cause antibody to develop if you transfuse a patient who is rhesus negative with the rhesus positive blood. So therefore, it is very important for us to, in most cases, and routinely detect weak D in our donors and the patients so that we give the right blood. And the same applies to the newborns, or if you're doing cord bloods. Yeah, so the weak D is detected in an indirect anti-human globulin test. So this is directly, in most cases, you refer this as Coombs test. Very few of us actually do this. Go ahead to do this Coombs test. So the Coombs test that I've always seen clinicians asking for is when they're investigating any transfusion reaction, if it occurs. That's when they ask for Coombs test, maybe direct and indirect Coombs test in some facilities. And yet we are supposed to actually be using this to confirm some of this. So for partial D, the rhesus antigen consists of many epitopes, like I mentioned, but very few of them are actually normal. Those few are able to actually stimulate the production of the what? Of the anti-D. Now, the partial D antigens lack one or more of these epitopes. So partial D6, especially partial D6, is particularly very important in transfusion because the D6 patient may develop antibodies if transfused with the normal D positive cells. This is very important, ladies and gentlemen. So we must handle this as D negative patient. Then the partial D antigens may react differently with the different monoclonal anti-D reagents. So it's very important for us to start opening our eyes about these cases of rhesus negative. The moment you meet a rhesus negative case, please interest yourself to go ahead and confirm whether that patient is actually rhesus negative or there is weak D somewhere. You confirm by doing a weak D what? Test. I have the procedure ahead. So what do we do with the weak D generally? Okay, so the true weak D samples react positively with anti-D reagents. Okay, but in most cases, you have to use the indirect anti-human globulin test, which is commonly known as Coombs test. The IAT, which is our indirect anti-human globulin test, relies on an antibody against human antibodies. Usually, the FC portion, this is not very relevant, but it's just good to know that you should be using anti-human globulin test to confirm your weak Ds. So let's look at the test methods. I have interested myself in only picking what can work for us, which in this case is our Coombs test or the indirect anti-human globulin test tube method. The gel is not really common in our facilities, so you can only use the Coombs test. And then for facilities that already have fully automated echo machines, I'm sure they're in regional referral hospitals and national referral hospitals, you don't have to hassle with the other tube method. Just use your automated platform to do your weak D, to confirm your weak Ds or do your blood groupings. For some high-end places, you can have other alternative analyzers like the Olympus. Yeah, but at least in our setting, I know we have echo, which is already placed in some facilities. So please make best use of them. But if you don't have echo in probably like Health Center 4, you can use the tube method to do your Coombs test. Yeah, so it still involves mixing your anti-D. You intubate your anti-D with the red cells in the tube. And then you add your anti-human globulin reagent, you centrifuge the tube, and then you read the reaction. So it is either positive or negative. And we know how to interpret this positive and negative. If there is an agglutination in the tube, then that is a positive result. If there is no agglutination, then that is a negative result. So let's go briefly into the standard operating procedure. I know many people at the end would ask, so how do we do this week D? It is a simple Coombs test. So what is the purpose of this? To actually guide the lab personnel on the procedure for confirmation of rhesus negative patients and donors in order to exclude or confirm the presence of weak D antigen. If it is there, definitely you will see the positive. If it is not there, it will still remain negative. So what is the principle of this? The red cells with the weak D antigens have a reduced amount of expression of D antigen, which I've already mentioned. So this requires using an indirect and human globulin reagent for the detection of the weak D. So procedure number one, what do you do? After doing your regular or your routine blood grouping, okay, and then one turns out to be rhesus negative. So you have to identify and pick it, okay, and the labeled 2 new test tubes with the sample number that is in tube 1, and then another tube should have the control number, okay, just to check your procedure. Then place 1 drop of the IgG and the anti-D, which is our reagent commercially prepared, into a clean label tube, test tube 1 and 2, and then add 1 drop of add 1 drop of the 5%, add 1 drop into the test tube 2. This is for the control, sorry. This is for the control. Number 4 is for control. Then add 5% red cell suspension of the rhesus D negative sample into tube 1. Tube 1, now you're dealing with that sample which initially turned negative. Now you want to deal with it to confirm whether it's a true negative or there is a weak D somewhere. So you're adding 5% red cell suspension, like two drops into tube 1. Then intubate the test sample and control tubes at 37 degrees for 30 minutes. It can have a range of 15 to 30 minutes, but it's very important to choose one. So 30 would do. So you mix it and centrifuge at 1000 revolutions per minute for 1 minute. And again, some people don't get very good results with the speed of 1000 revolutions per minute. You can choose to increase the speed to 1500. It gives you very good results as well. But the time remains 1 minute. Okay? So then you resuspend the cell buttons and examine for agglutination. So if there is no agglutination, what do? You proceed. You wash the red cells in the same tubes 1 and 2 3 times with a normal saline. And sometimes you can choose to use low ionic strength solution if you have. Even washing the cells 3 times, if you have the low ionic strength solution, you can choose to have this. This gives very good results. We've witnessed it somewhere where they were failing their EQAs. Okay? Yes, then you add 2 drops of anti-human globulin reagent in the test tube 1 and 2. And remember, 2 is the control. So you mix gently and centrifuge at 1000 revolutions per minute for 1 minute, and then gently resuspend the cell buttons. You examine for agglutination and grade them and record them. It can be +1, +2, +3, or +4. Then you had your check cells, which in this case we are calling it IgG-coated control cells. So this is the check cells, which should validate your reagent and then the procedure. So you add the check cells into tube 1 to confirm the validity of the anti-human globulin reagent and your procedure. Okay, so how do we interpret these results eventually? If there is agglutination in tube 1 after steps number 9 and 12, okay, which is addition of the anti-human globulin, it means weak D is positive. It is weak D positive. So it now deviates from your initial reporting where you said this was rhesus negative. But now if after adding anti-human globulin it turns out to be positive or there is an agglutination, so this is a weak D positive. If there is no agglutination, it means it is actually true rhesus D negative. So you comfortably report that as rhesus D negative. Agglutination after adding check cells validates your procedure and the reagent. If there is no agglutination, you need to repeat this. It means your procedure was invalid. And then no agglutination at any phase in the control tube invalidates the test. Okay, because remember you're using a non-control. So if there is no agglutination, then there is a problem with that. So you need to start afresh. Okay. So what is the summary of the clinical significance? Why are we hassling to make sure we do this WIKID test, by the way? So for the case of donors, especially us who handle donors, WIKD donors are classified as RESAS-D positive and can donate comfortably to the RESAS-D positive recipients. This is, you forgive me, this is recipients. Okay, the spelling is REC. Okay. Then for the patients whom you are handling at your facilities or the recipients who receive this blood, why is it significant to these people? Because the WIKD positive individuals are typically managed as RESAS negative to avoid alloimmunization. How about the pregnant women who report to your facilities for prenatal screening? Because blood grouping is one of the tests that is done to those mothers. Why is it important to confirm these RESAS negative? Because the weekly testing helps to determine the RESAS immunoglobulin, which is our ROGAM, the anti-D, which is given to our RESAS negative mothers. It can be in form of prophylaxis. So do they qualify? Are they eligible to also get the ROGAM or not? Because remember, for those who are actually true negative, the pregnant women who are RESAS negative, they must be given the anti-D shot at week 28 and maybe after 72, within 72 hours of after delivery, rather. So you realize that doing this or making confirmation of this is very helpful to these mothers, because ideally we are not supposed to just give the 500 milligrams, which is more like a standard in our setting. We should determine the degree of the negativity in this case. But in our settings, we can only do week D. We can't do other investigations. So there are some mothers who can actually qualify for these ROGAM prophylaxis because they are week D. Okay. Yeah. So what are the best practices and recommendations that we can take? Let us standardize our serological testing protocols to minimize these discrepancies. And then we also implement RESAS-D testing algorithm in transfusion laboratories. Any lab that is doing blood transfusion, can you make sure you have the protocol or the procedure for doing WIKD test? Also educate clinicians on WIKD management for transfusion and pregnancies. And by the way, some individuals, some mothers would have what is called fetal leakage at week 28. So they actually qualify to get the ROGAM at week 28. Others may not need it at week 28. They might need it when they have given birth within 72 hours. But let us also maintain the accurate records to prevent alloimmunization risk. For example, there are some cases that clinicians can, some decisions that clinicians can make about maybe a patient who is RESAS negative is a mature lady who was in her menopause, or an adult man who is also in her late age. They're all RESAS negative, but now they make a decision to say, okay, if we have looked around for RESAS negative and we've not found a RESAS negative anywhere, and if you don't give blood to this patient, he's going to die in the next 30 minutes or an hour. But we have a RESAS-D positive here. We can give that RESAS-D positive. That is a decision the clinicians can make. But the problem with this, or the trouble with this, is that for as long as you don't make that record known in the database of that patient, and then he receives the RESAS negative again the second time, there will be a transfusion reaction. Okay? But we cannot risk on young ladies in childbearing age to give RESAS positive to RESAS negative. No, this should not happen. But this decision can be made for those who are in their menopause, or some adult men, as long as you are going to write on their foreheads or their faces that I was once given RESAS-D positive, yet I am RESAS negative, which we don't have that checks in our Ugandan system. Okay, so it is risky. So what are some of the quality control measures, ladies and gentlemen? Number one is that we need to standardize our RESAS-D testing protocols. by the way, by implementing consistent testing procedures across our labs to ensure uniform results. What is done in Mulago should be done in Mubende, should be done in Lira, should be done in Kavale, should be done in Moroto, something like that. So that you also utilize the standardized reagents and equipment for accuracy and reliability. We already have some of these equipment, by the way. You are not, it's just a matter of showing interest that you can actually utilize the reagent once given to you. But if you give you equipment and then the reagents start expiring without you, or you're still running back to your tile, yet you have the equipment, it messes up with the standardization. So in this case, for those who already have the echo in regional referral hospitals, please make best use of them. It will sort you out with some of these discrepancies. Then we need to also embark on training laboratory staff on RSD typing methods. Which one works? So we have to provide regular training sessions, which this is one of the platforms we are using to update you with some knowledge for those who had never known. And also if there is any upcoming updates or technology, we bring on board and make sure you get trained. So I'm also, in my opinion, interested in making sure that the weekly test is part of the proficiency a testimonial that is, manual that is given to you. Okay, you don't only do ABO. If one reacts, turns out to be rhesus negative, you should proceed and do the weekly. So it should be part of the proficiency testing. Okay, and then the last but not least is continuous monitoring and validation of the test methodology. So we have to regularly assess testing procedures to identify areas of improvement and also validate the new techniques through proficiency testing and internal audits. I hope we do this. Many facilities that's actually enrolling for accreditation usually do these internal audits. So this should also be part of your internal audit. So maintain records again of the testing outcomes to track trends and performance, and still goes back to those patients who could have actually been given rhesus positive yet rhesus negative. Okay. So ladies and gentlemen, these are some of the references that we can get these resources from. And I want to thank you so much for giving me the audience. Thank you, thank you, thank you. Back to you, Chair.
Thank you so much, Mr. Kello, for this masterclass information that you've given to the audience. I really appreciate it. It was very informative, and colleagues also at the same time very practical. So I pray that we all go back to our respective facilities and implement this knowledge that Mr. Sam has given to us. To emphasize more what I've picked from him, really we have learned a lot. First of all, the structure of the rhesus D. We also validation of our reagents, of course, to obtain accurate results from our respective labs is key. And I think very soon from the blood bank perspective, we will request our regular supervisors to cross-check with the different labs that are accredited to transfuse to see whether they are putting this knowledge into practice. And that will form part of the accreditation procedure in the respective labs. Also, of course, to emphasize most is the use of the right methods that he has put across to us. So when the presentation is shared, you'll definitely follow through the details of how you can carry out some of this testing, but kindly choose the right methods. Also, from my point of view, I would like to request that any discrepancies of blood groups that you receive at your facilities, we request that we send the samples or get us to know about discrepancies of blood groups from the blood bags that we send to you so that we can resolve this and get the right records documented for the patients. So you can always call on us, or when you're picking blood, you send your discrepancy report and also some samples so that we can follow through with the investigations. Also on the side of the clinicians, of course, from that information we have got, it's very important that we get the right blood group for the patients confirmed before we put them onto the anti-D therapy, especially for the childbearing mothers, so that we don't put them at risk of alloimmunization. Also, to emphasize to the colleagues that do the testing and confirmation is we have to be cognizant of the pre-analytical, analytical, and post-analytical factors that contribute to perhaps wrong blood groups given to these patients. I know those that are implementing quality in their labs know about this very much, but also I would encourage all of us as practitioners to look into these factors: collection of samples, servicing of our equipment that we use regularly, validating the methods that we use in our labs so that we don't get errors forward. In transfusion, we believe in zero tolerance to errors as much as we can to avoid patient bad outcomes due to ABO transfusion reactions. We also encourage clinicians and also the lab personnel to rationally use blood. When we talk about the rhesus D negatives, we really face a lot of challenges as a blood bank to make available these products because there are quite few of them in the population, no more than 5%. So please, when you have your rhesus D negative patients, and please use the blood correctly. Do not waste that blood to patients that don't need it, so that we reserve it for the patients that really need it. We also encourage you to mobilize for us donors that are rhesus negative to come, because we have a registry for them. So when you send us emergencies, we're able to call these donors so that they're able to come and donate blood for these kind of patients. So we have a platform for the rhesus D blood donors, and I encourage all of us to mobilize people to register with us on that platform so that we can make available these products for the patients. So I would like to not waste much time. I would like to invite our next presenter, Lieutenant Robert Namisi, to start sharing his screen. But first, I would like to thank you, Mr. Okello, so much for the presentation. Please stay online so that we'll be able to be present on the Q&A session in a few minutes. So, Mr. Robert, kindly start sharing the screen, and then we'll move on to the next session. Colleagues, kindly register on the chat session. This is so that you can obtain your CPD points for all the professionals. It's approved. So kindly register. There's a link over there for you to register for your CPD points, which is very important these days. So kindly register on the chat for that as we move on to the next presentation. Thank you.
Yeah, I request that some unshares.
Okay, Sam, could you please unshare your screen? Thank you. That's nice. I hope everyone can see Robert's screen. Robert, you're most welcome for the second presentation. I would like to hand over to you to start your presentation. Thank you so much.
Very much, Chair, and good evening to members around East Africa, maybe a morning some other areas. Robert, as they have introduced me, most people know me as Masamba. It is part of my names. And thanks for this opportunity to share on this platform today. That is Umlta, that's why we are seeing that logo. But aware that I'm a member of the transfusion team, some presenting under UBTS, that's why I've also put the logo. But I work in UPDF, and a supervisor at the lab but resident in the blood transfusion section. Before I go in this presentation, we shall realize that it closely relates to what Mr. Sam has presented, especially when it comes to handling issues of weekly, because this is an automated procedure that will allow us to successfully do the weekly test. Secondly, he talked of validation. The method I'm going to talk about, you can't run samples without running controls. So that is also a big issue, which we shall see in the methods as we shall go through. And lastly, I would wish to emphasize that if you follow the Samuel's presentation, at a certain stage when you are intubated, we talk about washing cells. Now, first of all, how do you intubate when you are doing it on the time? Two, how do you wash the cells when you are using the tail? So at this point, we should be saying no tail. Okay. Our topic as it reads, embracing modern technology in blood transfusion. And specifically, we are looking at e-columnar because it is what we are using. It is placed at the hospital bases, regional and national facilities. First of all, before I go ahead, I would wish to declare a disclaimer that I don't have any conflict of interest in whatever I'm going to present. Now, the outlay of the presentation will follow the following. We shall have a brief definition of a columnar, the principle, mode of operation, advantages, factors limiting maximum output, improving output of a columnar, and then the conclusion. What is an columnar? This is a blood transfusion machine. It is a compact closed system and a fully automated system for blood testing. It is a microprocessor controlled instrument. It is an immunohematology in vitro diagnostic testing of human blood. Standard immunohematology assays using micro well strip based platform. On the left, on the right, you can see the machine we are talking about. That is the e-columnar machine we are using, which is placed in many of the government facilities. Another thing is that this machine is able to load samples anytime. Anytime you request for additional tests, it will keep on running. Now, this is a brief of, I was not able to get a video, but when you look at this set here, in the middle, the staff is trying to load the information of the procedures. Then these are the R capture strips where the reactions take place. They're also loaded in the machine. And here is the loading of the reagents. And that is part of the procedure when you are handling this machine. Now, we look at the tests being performed. Of course, the first one being the ABO grouping. It is able to do neonatal ABO grouping. We do rhesus D grouping, antibody screening, cross-match, weekly, which has been the topic today, which I expect that we shall put more emphasis, and direct antiglobulin test. Now, other versions which are bigger than the e-columnar, I think they're placed in the donor centers. They're able to do other activities, which include typing and others. Ours ends on the above tests we have talked about. Now, the principle, it has two phases. One depends on hemoagglutination. And when we're talking about hemoagglutination, we are emphasizing antigen-antibody reaction. And this is usually for red blood cell typing, that is ABO and ratius D typing and phenotyping. Then the solid phase, it is for antibody detection, identification assays, and compatibility testing, majorly focusing on the compatibility testing or cross-match. How it works? It is able to move the microwave strips, the liquid reagent fluids, and blood sample fluids to different processing areas. Now, when you are using the manual methods, at one stage you have to incubate, you have to wash the cells, you have to centrifuge, and then you go to the microscope if you use the tube method. But all these things are moved in this machine because it is a well set up system that everything is run in that machine phase by phase until you get your results. Now, once the procedures have taken place, then it captures the microwave images using the charge coupled device. In brief, it is called CCD camera. Now, this device is able to change light into electrical waves which can be read, and that is what we are able to read on the screen. Now, it also calculates the value of, for each well-based on multi-tutor image analysis using inbuilt software. Now, it assigns a result and interpretation to well-based predefined criteria associated with the calculated reaction. So all the process is done in an automated system. Yours is to feed in the reagents, feed in the samples, then you command all the procedures take place and you wait to read the results. Now, why should we go in for a columnar machine in blood transfusion? First of all, as we have heard from behind it, is that it performs all procedures in a one stand, placing and it continues. Assuming you are working in a lab with different equipment like ours, blood transfusion is together with hematology. So you run your sample in CBC, get it, put in the columnar. You can continue running other samples in the CBC machine as this one runs. Two, it emphasizes on running controls before testing. Without controls passing, the machine doesn't accept you to run the test. And that assures us the quality they were emphasizing. Three, in one of the samples you are using, it does multiple things in the shortest time. For example, when you are doing grouping, the grouping results will come out within 15 minutes after going all the phases. Then cross-match will take 30 minutes. So if you are doing grouping and cross-match, that would take around 45 minutes. If you compare with the manual method where you have to wash cells, centrifuge until you reach the microscope, that time goes beyond. And remember, there you will be working on one patient. But the other advantage with this is that each batch, as you press start, is able to run 20 samples as they go. Now, these 20 samples, depending on what you have got, but it can give you a variety. That the 20 samples, you are running blood grouping, you are running cross-match, you are running weekly, it accommodates all at a go. And it will read your results depending on what you have commended. Another advantage is that this machine uses tubes with different diameters. We talked of neonatal. You know, those kids are too small that we may not have enough samples, so they have special tubes. So you can use the ordinary vacutainer tubes for adult samples, but when we are handling those neonates, because of the samples we get, we use the neonatal tubes and it is able to accommodate. I've already talked about continuous loading without testing interruption. It keeps running as you keep loading samples, as long as there is space for you to add the samples. Now, for government facilities, reagents currently are passing through LMS. And this is for government facilities, I would emphasize. For private, I think the vendors of this equipment are on the line. They are training, they will tell us the procedures, how to access those reagents, but the company which delivers is based in Uganda, called Biomedics. Now, all those good things, what could limit the output? Here I was referring to why do we have low performance of the tests using this machine? One, you have to operate this machine when you have got a training. Of course, it is a short training when they're installing, but majorly it depends on the attitude of the personnel working on the machine. We are very much rigid to use the old methods, including I'm in the hospital, but even doctors will come and say, can you use that shortest method of ours, meaning the tail method? It is upon us to educate them that, please, this is not a safe method. Secondly, this machine in some areas where the temperatures are too high, those who have observed towards the end of February, the sunshine has been very hostile. So in such temperatures, if it goes beyond 35, the machine tends to go off. We experienced that last year and we got a solution for it. Next, you need an ideal and secure infrastructure where there is good ventilation, enough light, away from dust. But I was emphasizing that you can't do blood, you cannot be accredited to do blood transfusion when you don't have a room where you are going to do those services. This machine also is ideal for large volume facilities, say national hospitals where they have placed them, general hospitals, but we have some health centers which have high volumes, and the general hospitals like a case of I've ever worked in Iganga during my week, and I saw the numbers are really very high. But for private facilities, it is also highly recommended because this machine tells you the number of tests we have done, who has run it. So if someone has picked another sample outside, you will be able to account where that sample came from. But as I emphasized earlier, you can be running multiple equipment at a go, including the columnar. Then another issue is that the equipment requires other functional equipment like a centrifuge, because it is used, you first centrifuge that sample before you load in the machine to continue the procedures. How do